This is Brookings APBIO podcast about lipids. We talked in our last video PowerPoint about carbohydrates, and this is our second big macromolecule. Lipids are made mainly of carbon and hydrogen with a very little bit of oxygen. If you remember when we talked about monosaccharides, carbohydrates in our last slideshow, they have a ratio of 1 carbon to 2 hydrogens to 1 oxygen. Lipids have those 3 atoms again, but they're in a little different ratio. This is an example of a fat and is a kind of lipid. And notice lots of carbons and hydrogens, but very few oxygens in a lipid molecule. Lipids are a very diverse group. It includes things like fats, steroids, phospholipids are kinds of lipids. And lipids are unique in the big 4 macromolecules because they're the only ones that don't form polymers. They're also the only ones that are very phobic because they're long hydrogen-carbon chains where we don't have a polar. Remember, these are non-polar kinds of connections. Lipids, fats, are very hydrophobic. They don't form long chains made of similar subunits. They're not continuous chains like we saw. Remember, think about those pop beads we talked about, making little subunits into a bigger polymer molecule, like when we made polysaccharides. If you look at this fat, there are pieces that go together. We have 3 that are similar. These are what we call fatty acids, long hydrocarbon chains, and they're attached to a smaller little molecule called a glycerol molecule. Notice though that it's not one long continuous chain, and there are actually 2 different kinds of pieces that go together to make a fat. Fats have a structure with a kind of a backbone molecule called a glycerol, 3-carbon molecule. It's a 3-carbon, notice the alcohol groups, the OH, hydroxy groups, and joined together with long, very long hydrocarbon chains called fatty acids. What makes it an acid, a fatty acid, is this group at the end. Remember those functional groups we talked about in our carbon chapter? It's got a carboxyl group at the end of the long fatty acid chain. And that allows it to do that dehydration synthesis reaction. I can remove a water molecule and hook that fatty acid chain onto one of the carbons here on this glycerol. And notice there's a couple other carbons that I could add fatty acid chains to. And we're going to use that dehydration synthesis, pull out the water and make a bond. Fats are called triacyl glycerols. There's tri for 3. There's 3 fatty acid chains attached to that glycerol molecule. And there's a linkage, this is called an ester linkage, that holds those fatty acids to the glycerol backbone. Fatty acids can be used for, that we put on our fat can be the same. All 3 of the fatty acids joined onto that glycerol can be the same. They can be different, different lengths, different numbers of carbons, different locations of double bonds. They can be saturated. All the hydrogen carbons are completely full with hydrogens. We call that a saturated fat. Or it can be an unsaturated fat. Remember we talked about double bonds making carbon molecules unsaturated. If you have an unsaturated fat, because of that double bond, it puts a little bend in that fatty acid chain, a little kink. And wherever you have a cis combination, remember we talked about cis and trans isomers across a double bond. If you have a cis isomer that is a hydrocarbon chain, it makes the molecule bend a little bit, and that's important in how it goes together in making that molecule. What do we use lipids for? There's energy storage molecules. very rich in energy. They have more energy stored in a same amount of same mass of fat as you do in a carb. It cushions your organs. It insulates you for body temperature. Think whale blubber, animals that live in very cold places like whales, walruses. They store fat in their body to help keep them warm. Saturated fats are long hydrocarbon chains where all the carbons are bonded to hydrogens. There's no carbon-to-carbon double bonds, and they make long straight chains. Animal fats, most animal fats are saturated fats, and we talk about saturated fats in our food. These, because they make long straight chains, they pack together very nicely, and they act as solids at room temperature. Think butter or lard. And they, too much saturated fat in your diet can lead to plaque deposits in your blood vessels, cardiovascular disease, talk about atherosclerosis. We have to kind of be careful of the kinds of saturated fat that we're eating. Unsaturated fats have double bonds in the fatty acid chains, but it causes a little kink. Remember the cis, we talked about cis and trans. If it's a cis isomer where both of the hydrogens across the double bond are on the same side and the rest of the hydrogen carbon chain is on the other side together on the double bond, it forms what we call a cis isomer. And because of that kink, those molecules, the fatty acid tails, don't pack together quite as tightly. So most unsaturated fats are liquids at room temperature, and we find them in plants, vegetable oils, fish oils. That kink prevents the molecules from packing so closely together. The other thing that we do with fatty acids and a kind of lipid that's very important for cells is making phospholipids. We learned about phospholipids in bio. Has that kind of unique shape with a head and long tails hanging down. The tails are made out of hydrocarbon fatty acid chains, and the head is a polar head. That's where we find the glycerol backbone and the phosphate group. That's important for giving that head its polar charge. Remember, phosphates are negative. If you have a phosphate, it makes that molecule, that part of the molecule, more negatively charged. So notice now we're going to use our same glycerol backbone, that 3-carbon little alcohol molecule, and this time though we're only going to hook on 2 hydrocarbon chains to 2 of the carbons, and the 3rd carbon gets the phosphate group up on top here. Why are phospholipids important? They allow the cell to make compartments, to define areas, to say inside, outside. Phospholipids, remember, because they are lipids, are very nonpolar. They don't like water. They're very phobic. And think about if our cell membranes were polar, they dissolved in water, we would not be able to make a barrier, make a compartment. If you look at how phospholipids go together to make cell membranes, remember the tails are very phobic, and so they orient themselves so that those phobic tails make a sandwich and they stay in the middle away from the water. Think if this is a cell membrane, there's water on both sides of that membrane, inside the cell and outside the cell. And so the heads, the polar part that likes the water, lines up on the outside like the bread of the sandwich, and the tails that are very phobic line up on the inside away from water. And that's important in making cell membranes. That phobic tail hides from the water, and because of that nature of wanting to be phobic, if you put lipids into water, they will automatically form shapes that allow the phobic tails to not touch water. So we're going to watch a little animation. If I take phospholipids and throw them into water, they will orient themselves. Notice the heads like the water, but tails are trying to stay away from water. If you look at how they line up, polar touching the water, phobic tails away. If you shake them up, sonicate them, what happens is you can actually form these little spheres called micelles with the tails on the inside. You gotta think 3D now. This is sliced so you can look inside. All around the outside are those polar heads touching the water, and the tails face inward, stay away from water. If you add more phospholipids, and then it makes it so it's harder and harder for them to orient the way they want, they can actually form a double layer, a boundary, with the tails in the middle like a sandwich, heads on the outside touching the water, and basically that's a cell membrane, right? It's a bilayer. So micelles, bilayers are all arrangements that form naturally when phospholipids are trying to, the tails are trying to stay away from water in the environment. So phospholipids and proteins are important components in cell membranes. We learned about that in biology, and the phobic-philic nature of that molecule, because it is a combination, both phobic and philic in the same molecule, it allows those phospholipids to make barriers, to make cell membranes, make it so we can have compartmentation in cells, and it's a very important shape-function relationship. The other kind of lipids are steroids, and steroids have a little different shape. Instead of very long hydrocarbon chains, they form rings. And remember, at the corners of each of these little hexagons, this is a glucose, a sugar molecule, and carbon, 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 carbon. Basic steroid is cholesterol, and anytime you see this 3 hexagons with a little pentagon, five-sided shape, it's a kind of steroid. 4 fused carbon rings. And what makes this different steroids different, we start with that cholesterol building block, but we can add different functional groups. And remember, functional groups added to molecules give them different properties. So for example, different kinds of steroid hormones, think testosterone, estrogen, are kinds of steroid hormones. They're reproductive hormones. Cortisol, aldosterone, they're different kinds of hormones. And depending on what groups you add to that basic body shape, you can have different kinds of molecules doing different things. So cholesterol is our building block for making those sex hormones. And changing those functional groups can make a big difference between, look at the difference between female hormone and the male hormone. One little functional group, adding a hydrogen here instead of a double bonded oxygen, makes a big difference in how that hormone acts in your body. Cholesterol is important. We talk about, often hear about cholesterol being bad and we don't want cholesterol in our body or in our food, but cholesterol is an important building block in making those hormones, those steroid hormones. It's important in cell membranes. Animal cells have cholesterol in their cell membrane, and it's that precursor for those other sex hormones. However, too much cholesterol can cause cardiovascular disease. It can lead to plaques in your blood vessels, and so we have to kind of watch how much cholesterol we're getting in our diet. That's the end of lipids, and stay tuned for proteins coming in the next slideshow.